Thank you everyone for being here, and thank you, Dr. Chatterjee, for being here for this last seminar talk of the fall semester. Dr. Chatterjee is an assistant professor of bioengineering, computers, and information science at University of Pennsylvania. His Programmable Biology Group develops new generative models for novel design of peptide and protein biologics, spanning from algorithmic theory to preclinical validation in vitro and in vivo. The lab specifically develops therapeutic interventions for rare neurodegenerative diseases, pediatric sarcomas, viral infections, infertility, and substance use disorders. Having received his SBSM SM and PhD from MIT, Dr. Chatterjee has received the NIH MIRA Award, Hartwell Individual Biomedical Research Award, and multiple NIH and foundation grants for his work. He has also co-founded Gameto, UbiquiTx, and AtomBioworks, which translates his research into fertility-related solutions, cancer therapeutics, and RNA medicines. And I'll hand the floor to Dr. Chatterjee.
Awesome. Thank you, Vivian. Thank you all for being here. I hope on this Monday you're excited to hear some news stories from our lab on the design of programmable biologics with generative sequence models. So as Vivian mentioned, I'm a professor of bioengineering and computer science at Penn. And I always like to start by showing the lab. So half the lab are pure AI scientists that develop generative algorithms. And these algorithms we utilize to develop protein therapeutics that my experimental team can go into the lab, do translational work, and hopefully make medicines. And so as you can see, our work really spans a wide variety of fields from very theoretical and mathematical frameworks through applied algorithms, protein biochemistry, all the way to in vivo disease models. So the proteins that we create, though, are what we call programmable. What do I mean by that? Well, we first develop proteins that can edit any DNA sequence, for example. And you guys probably know these proteins. These are Cas9 enzymes for gene editing. So over the years, we've developed bioinformatics algorithms that can identify the DNA binding specificities of various CRISPR proteins. And we've used SPAM a lot to engineer and discover broad targeting CRISPR enzymes like iSpyMac, SC++, and Spryce, which we've utilized to fix mutations in genetic diseases that were previously inaccessible, such as Rett syndrome and Huntington's disease. We also develop proteins that can generate new cells and new tissues. We've created graph theory algorithms like StampScreen, which can identify key transcription factor nodes in the graphs of gene regulatory networks. Those are proteins that we can then put into stem cells and convert them into ovarian cells. These are the cells we're interested in. I started a company on this called Gameto, where we take immature eggs from female patients, we co-culture them with these cells that we've generated, these ovarian support cells, and that allows us to mature these eggs into fertilizable M2 oocytes. And this is in the last year, we've actually had many babies born from this technology. Baby Mia is the first one in Peru. We actually had one born in Australia this week. So Gameto is actually the first US company to achieve a phase 3 with a stem cell product. So it really just goes to show you that this functional algorithm to protein strategy, I think, works really well. And so the rest of the talk, though, I'm going to talk to you about biologics that we are developing with generative models that can bind and modify other proteins. Because look, it's not DNA or RNA that cause disease. Primarily, it's proteins. And if you look at all of the cancer-causing proteins that we have prioritized, over 300 of them are considered intractable via standard small molecules. They don't have binding pockets, so that you cannot design drugs very easily or at all, really, to these targets. Moreover, a lot of the proteins look like this. They're very disordered. They don't have conformational stability. And so if you're going to use structure-based methods, your standard crystallography or cryo-EM strategies or AlphaFold, those methods will not work. Models that have been trained on structural priors will not be ideal for these proteins. So instead of small molecules, let's go to the next best thing, which we think are peptides. Instead of structure, let's think about methods where we don't have to go through a structural prior. So the question we've asked is, can we design peptides that bind and degrade undruggable targets without needing to use structural information? And so our first instinct was to train protein language models. You guys are very familiar with language models like Claude, ChatGPT, and Gemini, but these models are trained on millions and millions of text examples, and they're not annotated. It's not like this is a happy text, a sad text. This is the noun, verb, adjective. And the same can go for proteins. We can just train on sequences, no structural priors, to learn the implicit structure, function, and interaction of these molecules. And this is simply by using a standard mass language modeling task, which is a classic representation technique for AI. So we can take this pre-trained language model that we've trained on protein sequences, and we can say, okay, here are where real peptides, these smaller proteins that we like to design, this is where they exist, right? Here are the sequences. We embed them in latent space. This is the area of the latent space they exist at. Let's take their representations. Let's apply Gaussian perturbations to those representations and now decode those numerical representations back into the valid peptide sequences that we can generate. These are new peptide sequences. They're not the existing ones, but they're similar and they have good physicochemical properties. We can then screen them for binding to our target by training a separate language model. We call this a contrastive model where we can see if a peptide and protein do bind, their contrastive score, their cosine similarity should be very high. But if a peptide and protein don't bind, we've trained the internal representation of this model to separate them. So this is very similar to contrastive language image pre-training developed by the OpenAI team. So this is called CLIP. We decided to create a peptide prioritization with CLIP pipeline using this algorithm. So we call this paperclip. And the idea of paperclip is really nice. We show that when we generate peptides to new targets, we can have these peptides bind with nanomolar affinity to their targets. You can see for beta-catenin, we get about 100 to 200 nanomolar affinity, pretty good binder. Then we can show that by testing about 20 peptides, we can find multiple, actually 19 of the 20 we tested on the left here in blue, we're able to inhibit enzyme activity by binding to this very structured enzyme called Ultra ID. It's a biotin ligase. On the right, this is RF diffusion, which is David Baker, who won the Nobel Prizes primary algorithm. They use structure, and still they do worse than paperclip on designing binders. We can then do something cool. We can say, instead of just binding, what if we attach these peptides to the catalytic domain, right, the enzyme domain of E3 ubiquitin ligases? This allows us to not only bind to the target, but also to degrade the protein. And as you can see, we now see degradation or removal of beta-catenin in this example, where we tested a couple of peptides from paperclip. You can see not only do their beta-catenin levels go down, we also see the corresponding signaling pathway, the Wnt signaling pathway, also go down. So this really is cool to show you that just a sequence model can not only induce degradation, but also functional effects. But the big kicker would be, what proteins can we do with this? Can we target with this algorithm that could previously not be targeted? And I point you to this protein. It's SS18SSX1. It drives synovial sarcoma. It's probably one of the most disordered proteins we know in nature, and it is the primary driver of the cancer it causes. As you can see, just we sent 10 peptides to our collaborators in San Diego, Ani Deshpande's lab, who have exogenous reporters of SS18, SSX1 activity. And if you see Paperclip 4 out of just 10 peptides was able to degrade this M-cherry fused construct. And to confirm that this worked, we took that degrader, that peptide, we put it into synovial sarcoma cells, and we saw complete ablation of this protein. You can see the poly A, which is the untargeting control, and you can see with Paperclip, we were able to selectively remove the fusion oncoprotein. By the way, the paperclip was done in a very generative plus prioritization pipeline. You make peptides, then you score them with a clip model. But you can also do this conditionally, where you can just give the target protein. We attach the binder sequence at the C terminus, so the right side of the sequence. We then hide the binder completely, and we fine tune a protein language model that was previously trained on single protein sequences now to unmask the binder sequence. It's kind of silly almost. And this fine-tuned version, meaning we updated all the attention weights and the MLP weights for the base transformer, ESM2, to create PepMLM, and this fine-tuned variant of ESM2 that generates peptides, you can say, hey, here's a new target protein. I want to design a 10 amino acid binder, and the model will literally just unmask that sequence. And so that's really nice because then you don't have to go through structure and you can just enter literally type in a sequence and the model will produce amino acids that it predicts to bind. This works. It sounds silly, but it works. We've tested a hit rate, and as you can see, over 200 targets we tested, 40% of papamolan peptides bind better than an existing peptide to that target. So this is a very nice controlled study. As you can see on the right, this is all structured proteins, so very stable proteins. RF diffusion, again, David Baker's best algorithm, which does really well. It has a 30% hit rate, but again, they use the structure, and just with a silly little language model, we can hit a 39%, 40% hit rate. As you would expect, these peptides do bind well. They bind with nanomolar binding affinity. They also are very specific. They don't have off-target interaction with, say, like BSA, which is a very sticky off-target protein. And we wanted to see if these PepMLM derived peptides, these binders, are able to actually mediate important interactions. As you can see here, if we take the mutant Huntington protein, which has a poly-Q extension via CAG repeat extension in its DNA, that mutant protein can degenerate neurons. And so we want to be able to target this mutant Huntington protein. We sent 5 peptides to our collaborators, Ray Chawant's group, put them in these degrader, these ubiquibody architectures, as we call them. And you can see we were able to completely ablate the Huntington protein in a Huntington cell model that they have developed. And some, I mean, this is all of them are degrading, and some are completely ablating the protein. Again, a protein that was previously considered undruggable by drug developers. You thought COVID was bad? Wait for Nipah virus or Hendra virus or HMPV. These are future pandemic viral proteins. We were able to design 20 peptides for each of them. And as you can see, multiple of them, in HMPV at least here, you can see multiple of them degrade this phosphoprotein that drives infection a lot, right? Over 70% degradation. We then took those degraders, we put them into live infection models of HMPV. And here on the left are viral proteins stained with GFP, and you can see the proteins completely really go away just because we were able to bind to this previously difficult to target protein. If you want to try out PepMLM, I know a lot of you probably are not at the experimental level, but if you do have experimental colleagues, tell them to try it out. We got about 4,000 downloads last month. It's very easy. You just have to type in a sequence. There's no like folded with AlphaFold, enter it, like wait 20 hours to get it to design. It's very quick. But here's the question. I've told you about proteins that we want to get rid of or we want to inhibit. But what if a protein is improperly degraded? For example, you may know that tumor suppressor p53 in its wild type state is a really good protein. It prevents tumor progression and proliferation. And so sometimes in cancers, wild type p53 is hyperubiquitinated by its cognate E3 ligase, MDM2. So what happens as a result is p53 levels go down, and that's something we want to avoid so that we prevent cancer progression. So can we stabilize proteins like p53 by removing ubiquitin chains rather than just adding them on? So we created a D-ubiquibody. Remember, we had ubiquibodies for peptide E3 ligase fusions. These are peptide D-ubiquitinase fusions. We use the OTUB1 domain. And as you can see now, when we treat beta-catenin expressing cells with our D-ubiquibody, you can see now the levels go up because we're stabilizing this protein. And we can then take algorithms like paperclip, make d-ubiquitous with them. As you can see between the empty plasmid or the control, we can see that the level of Wee1 significantly goes up. We've shown that PAPMLM-derived ubiquitines can stabilize very key proteins like tumor suppressor p53. Here's the non-targeting control. Here's the one treated with our dubiquitine. You can see the p53 levels going up. But more importantly, we can take these dubiquitines, we can put them as mRNA into a lipid nanoparticle, similar to your COVID vaccines, and we can deliver these into cells. And as you can see, now not only do we get p53 levels going up, which you'd expect, but we also got significant upregulation of apoptosis, which is what p53 enables. So again, not just direct functional stabilization, but we also get a downstream function as well. So I've shown you that we can programmably degrade disease-relevant proteins with these peptide E3 fusions called ubiquitines. We've also can programmably stabilize disease-related protein with d-ubiquitines, as I showed you. But now we're working on peptide retargeted phosphatases. So it's peptide attached to a phosphatase domain. We call these PepReps. And PepReps we've shown to induce tau dephosphorylation. So now we can remove hyperphosphorylated tau phosphate groups to be able to then mediate a therapeutic effect. We also have created PepDACs, which are peptide-guided deacetylases. So we're using the HDAC4 catalytic domain to just programmably remove acetyl groups. And as you can see, we can enable deacetylation of p53. So I would call this kind of a CRISPR system for proteins. The peptides are like your guide RNA and your enzymes are like your Cas9, but instead of editing DNA, we're editing proteins. By the way, we're translating these technologies in vivo. You can see that we've been able to degrade beta-catenin in vivo in mice by delivering these as lipid nanoparticles. And I've started a company called UbiquiTx that is translating these degraders, hopefully into the clinic or utilizing these as drug development tools. So hopefully I've proven to you so far that sequence is enough. We don't need structure. Honestly, I think structure hurts us to generate potent binders to different target proteins. The question is, can we make these peptides more specific, right? It's fine to just bind to the protein, but one, can we bind to specific spots on a protein? And to do this, we've developed an algorithm called MOPIT, which is a motif-specific PPI targeting algorithm. As you can see, Moppet relies on this previously trained classifier that we've designed called bind evaluator, which can take in a binder sequence and a target sequence, process their representations through different embedding layers. We can then interpolate these embeddings via reciprocal multi-headed self-attention to like connect the embeddings together and then use those joint representations to then go and predict on the right the binding sites on the target protein that the binder binds to. And if we use Bind Evaluator to guide the generation of peptides that bind to the specific binding sites you want, we're able to make very specific peptides. And as you can see, Bind Evaluator, just the model itself, achieves very high accuracy and AUC on predicting binding sites. That's a hard problem. And now we have very accurate predictors. That helps us to make these binders. By the way, I will tell you how the generation works in a second, but we'll get to it a little bit farther later in the talk. But I just wanted to show you quickly that Moppet is able to produce very high affinity motif-specific peptides that bind to the FN3 domain of NCAM1. As you can see, if you have binding to NCAM1 by itself, it works, but you can also get full-length NCAM1. That tells us that FN3 is where those peptides bind. We also can generate peptides with very specified motifs on targets. This is a protein called AGR2T. It is a protein that's known that if you bind to AGR2T, you recognize this, you can activate a CAR, you can activate a potential CAR, and this protein will allow us to then specifically get CAR Ts to different targets of interest. So we want to bind specifically to AGR2T. So we use MOPIT peptides to design to this protein. You can see they bind pretty well, but when we actually put these into CAR T cells as the binder, not an scFv or an antibody, you can see we can activate the CAR. You can see with M2, our peptide, you can see the activation marker goes up. So we're now with non-targeting CARs, we don't see that activation. So yeah, we've made CAR Ts with motifs targeting generative models. We are also working on PTM specific peptides. And this is something that we are still in progress, but we've created a language model that was published this year in Nature Methods that actually just incorporates post-translational modifications. It's kind of crazy that for years we've never modeled PTMs. We always just model wild type sequences in protein design and modeling tools. And so what we said was instead of just having the wild type protein sequence where you may have a tyrosine at position 5, let's add tokens to our language model that incorporate the PTMs. This is phosphotyrosine that we're incorporating at position 5. And we trained this really interesting model. We actually developed this algorithm that utilize this, what we call a state space model. You don't have to worry about them. No one uses them anymore. But the idea was we created a bidirectional state space model, a bidirectional Mamba model that can process both wild type sequence representation, just the regular amino acids, as well as the PTM representation, and we merge them together via some gated embedding fusion layer. As you can see on the right, there's a reason why we use Mamba. People ask us, did you just use it because it's cool? No. If you can see on the left, in training, you can see the accuracy and training of this language model converged much quicker than the regular BERT transformer model, and we actually ended up with higher accuracy anyway. So there was an argument for using this model. But the best part is now, if you have a protein that has a phosphorylation on it, you can see the difference between that and the wild type, right? Here's the embedding of the wild type and the PTM sequence. We just do this in low dimensional TC space. But the point is, it's not the same. Previously, for years, it was just the same. You had a phosphoprotein and a wild type protein. These models treated them as like they had no differences. But in reality, we know phosphorylation has an effect. And the beauty is also you can say, hey, here's a protein sequence. Here is amino acid 309 that has a threonine. What PTMs could be on threonine? You can mask and unmask that sequence, and the model will actually produce a logit probability across the PTMs. In this case, it thinks phosphothreonine should be at position 309. So it's a zero shot PTM predictor, which is super cool. Next, we're developing the PTM specific peptides that can say, let's just bind to the phosphoprotein and not the wild type, now that we know the differences between those two proteins in high dimensional space. We're also working on fusion oncoproteins. You may be like, what the heck are these proteins? But these are really cool, and they're not cool, they're kind of bad. But they're basically caused by a genetic fusion of two genes that are normal, but when they fuse, they create this protein, a fusion oncoprotein, that is completely new. The body has never seen it, has no idea what to do with it. And because these are usually transcription factors, they're really driving downstream signaling and in an unregulated fashion. And as you can see, these drive a lot of different sarcomas and most pediatric cancers, and they're very, very big and disordered. So this is the one of the best target classes we know in drug discovery, because if you degrade this protein, you cure this cancer, like bar none. But they're so weird, they're so wild, disordered that no drug company would ever think of trying it. And so we said, let's try to make proteins that drive, and let's try to design molecules that will be able to inhibit or potentially degrade these proteins. But the question is, it's not enough to just bind to the fusion oncoprotein. We saw previously with SS18, S61 that we were able to degrade it. But what we need to do for a oncoprotein like, say, EWS-Fly, we need to bind specifically to the fusion, but not touch the parts that make it up, EWS and Fly1. Those are actually important transcription factors. If you degrade those, you're going to have a lot of off-cell toxicity. And so Sophie in the lab has created huge amounts of information and models to be able to solve this problem. She first created an entire database of fusion oncoproteins, which didn't really exist, curated from multiple different sources to create Fusion DB. Then we trained a language model just on fusion oncoproteins. And that language model we've shown allows us to predict properties, because it's a representation model, to predict properties of puncta, nuclear localization, cytoplasm localization. We published that this year in Nature Comms. But the big goal is to be able to generate binders that are specific to the fusion oncoprotein. And so to do this, we have two models separately that do this, a specificity guidance module and a generative model. And we'll talk about this later, that is able to guide the generation to be very specific. So what is this specificity guidance module? By the way, the name we came up with is called SOAPy, which is specificity guided generation of off-target avoiding protein interactions. And SOAPy is by Sophie, so I don't know. Anyway, interesting name. But we take that. So how does this specificity guidance module work? As you can expect, we have to jointly embed the binder, the target, and the two off targets, which are your head and tail proteins, in a single latent space, such that we can say, let's distance the binder and target from each other, let's keep the binder and target close together, but distance the off targets away in embedding space, such that we know to score what sequence binds to my target specifically. And we used this is a Siamese model. So naturally, a Siamese model takes in three inputs and trains with a triplet loss, but we decided to extend this to a quadruplet loss to incorporate the second off target. And as you can see in training, the distances of the off targets in the pinks go up, but the distance to the target stays pretty close. So that's really nice. We know we're getting specificity good prediction. And what we then did was in Soapy, we added affinity to create Sophia because Sophie's actual name is Sophia. And we showed that by adding both affinity and specificity in our generative model, which I'll talk to you about in a second, we're able to get both of these incorporated and we get binders on the left. You can see these are the fusion oncoproteins. We get good binding scores. On the right, these are the off targets, the head and tail proteins. You can see we get low binding scores. We actually are testing these experimentally in the lab, and we're getting really cool isoform specific results. By the way, we don't have to work on just protein targets. We're also interested in targeting pollutants like plastics, also heavy metals, which we see pollute our environment, cadmium, cobalt, copper, lead. So these also create a lot of health risks. So heavy metals can create nephrotoxicity, genotoxicity, skin toxicity. So if we were able to design peptides to sequester heavy metals, we can make these either into therapeutics or we can use these in the field to remove them and clean up the environment. So to do this, we've created this model called Metallurian, which is a co-evolving diffusion framework of peptide binders. The way it works is we have a pre-trained classifier called Metallatte, which, as you can see on the right, really accurately predicts metal protein binding to different heavy metals, lead, vanadium, plutonium, nickel. We can get really good AUCRCs across this model. We can read about Metallatte. But the beauty of Metallatte is that we can now take its representations, which knows interactions of metal proteins, we can apply Gaussian diffusion on the continuous embeddings, right? The latent embeddings, but we can also apply categorical diffusion on the one hot label of the metal that it binds to, right? So one could be lead, for example, and so we are going to be able to apply categorical diffusion to that. The generative process is really nice. It's what we call a co-evolving generative process, where the reverse process, the goal is to create the latent embedding back, which will generate the sequence, but ensure while doing this denoising process on the categorical labels, we ensure that the categorical labels corresponds to the metal we want to bind to, in this case, lead. So this is a really beautiful theoretical framework that led to lead metal binders. And as you can see, we've done MD simulations first, just as a validation that shows that our peptides can interact with cadmium, with copper, but more importantly, we've actually built up an entire experimental system in the lab using the sandwich ELISA strategy that Divya, an undergrad in my lab, has spent a year developing, and she has shown that these Metallurian designed copper binders and zinc binders work very effectively. We're actually working to design binders to nickel to sequester it in cows that produce methanogens that depend on nickel as a substrate for reduced methane production. But look, even if we can get more specific to these target proteins, they still need to become good therapeutics. right? So think about it. The GLP-1 are agonists. I'm sure a lot of you have heard of them, Ozempic, Wegovy, Mounjaro. Did you know that they've been around for decades, right? The first exenatide and liraglutide, they actually were discovered in the 80s. Like I always say, this is well before I was even
Excuse
born.
me. Can you slow down just a tad? Thank you.
Yes, of course. Sure. Yes, of course. So no, of course, I'll be slow. I think we have a good amount of time too. So hopefully you've seen that we've been able to design these peptides, but what I'm trying to make here is that these peptides have been around for a long time, right? So if you look on the left, in the 80s, these GLP-1 peptides were discovered, but it only took until recently that these peptides like semaglutide were approved, right? 2021. So what's taken so long? The answer is that it took years to perfectly adjust these peptides to get optimal therapeutic properties, right? So as you can see here on the right, these are the peptide sequences, and they're no longer just wild type sequences. They have non-canonical amino acids. They have these large lipidation tails that had to be added for these molecules to be able to have the long half-life so you don't have to inject yourself every day. This is the point I'm trying to make. Therapeutic design is inherently multi-objective. So we need to also obviously bind to our target protein. We need the proteins to be soluble, or these peptides to be soluble. We want them to be orally bioavailable. So if we take them by mouth rather than getting injected, we want the peptides to have a long half-life so they don't crash out of our bloodstream. We don't want them to stick to random other proteins, and we want them to be non-toxic, right? So we want these to have multiple properties simultaneously optimized so that we have a good chance of these molecules going through clinical trials. The problem is current generation frameworks don't support this. If you look at models like ChatGPT, these are zero objective guided generation. They are just, you just give it a prompt and it's just predicting the next token. There's no objective. The prompt isn't an objective for the model. It is just an infilling. You're basically starting its answer, and the next tokens that it predicts are just extensions of that answer. While models like DALL-E or Sora, I'm just using these OpenAI as an example, these are what we call single objective guided generation models. So the generation of the image or the video is conditioned, as you can see here, on the text representation, right? So there's one objective, the text. But what we need is multi-objective optimization. And we need that for the molecules like peptides that we're designing. And to do this, we've developed an algorithm called Peptune. And Peptune is really beautiful. It is a model that allows us to say, given an indefinite number of properties and predictors for those properties, can we use that to guide the generation of peptides that bind to the targets of interest? As you can see, we don't utilize wild type sequences anymore. In this case, we're utilizing a chemical representation that allows us to model both regular and modified peptides, peptides that have methylation side chains or side chain stereochemistries or cyclization or non-canonical amino acids. To do this, we use a tripeptide structural representation that is a chemical smile. You can see no longer just like alanine, threonine, tyrosine. You can see that these are smiles, and we can then tokenize these smiles based off the side chains, the peptide bonds, so that we keep intact structures for these peptides. Great. So we can make smiles. How do we generate these peptides? To do this, we've developed an algorithm called, and we've built on an algorithm set called mass discrete diffusion. You may have heard of it. So any of you have heard of like Gemini diffusion? It's like this, basically Gemini is going to use this algorithm. The idea is actually quite straightforward. If you look on the left, here are smile sequences, or you can use letters or whatever you want. Over time, right, at different time steps, we corrupt that sequence until all the sequences are, or about all the sequences are masked, right? They have a mask at each of those positions, they're hidden. The beauty of Peptune is that we don't just mask at a uniform rate, we actually, or some like standardly increasing rate. As you can see, we actually reduce the masking rate on the peptide bonds, these are the ones that are in purple, we can mask those at the lower rate because guess what? If we don't have peptide bonds, you don't have a peptide. So we don't want to overly mask those sequences because in the reverse process here on the right, if you look, we're able to then regenerate over time steps the amino or the tokens, the side chains or the peptide bonds that put together the sequence. So at the end, we get a new peptide sequence smile, so we reconstruct it. If the peptide spile is not valid, meaning it doesn't have a peptide bond, we don't have the amino acid, we actually upweight the loss on the models. So we add an invalidity loss that allows us to make sure that we're reconstructing valid peptides from scratch. But this is just wild type. This is just peptide sequence generation. As you can see, though, it's good. We generate good peptides. They're permeable. They have binding data. They have binding. These are cyclic peptides. They have non-canonicals. But the question is, we want to identify a set of Pareto optimal peptide sequences across the properties that we're interested in. Membrane permeability, target protein binding affinity, whatever. We want these sequences that are optimal across all of these properties. So to do this, we developed an algorithm called Monte Carlo tree guidance. And the idea is we can instantiate masked discrete diffusion where we're unmasking at different time steps, right? Different time steps as a Monte Carlo tree. So in the expansion part of the tree, we're unmasking the tokens so that at the leaf nodes, the bottom nodes, we get a fully unmasked sequence. We can then take those fully unmasked sequences and we can score them with pre-trained property predictors for cell membrane permeability or target protein binding affinity or other properties, as many as you want, right? And what we do is we create the score vector here, which then scores them on each individual property of interest. Some of them are good, some of them are bad. But the point is we can compare how this peptide sequence compares to another one or to a previously scored peptide sequence on each of these scores, on each of these properties. If you have a sequence that dominates, meaning it's better on all and in all the properties, then we will upweight the path that created those sequences, meaning we looked at that path and it created a good sequence. So why not search that path again while generating? So that because then we can get better and more Pareto optimal sequences. And so then in future generations, future iterations of our MDLM, we're able to select the paths that create these Pareto optimal sequences, right? It's just an iterative inference time generative model that utilizes the initial generative model as a prior. As you can see, these peptune peptides can generate and simultaneously optimize peptides for an indefinite number of properties. You can say binding affinity to target 1, binding affinity to target 2, solubility, hemolysis, non-fouling. As you can see, they all go up, meaning we're optimizing across all these properties at the same time during generation. So that's nice because then it's no longer about binding. We actually have other properties incorporated. We've used this to create new GLP-1R agonists. So these are better than semaglutide or liraglutide. We've actually also designed peptides, as you can see here, that block this protein AMHR2. And AMHR2, as you can see, the peptides we've created for it are cyclic. So they're no longer a linear little peptide. They're cyclic peptides, and they have optimal properties across a lot of the properties we're looking at. We can also create binders that bind to two things at the same time, transferrin, GLAST. These can help us to get into astrocytes for specific diseases. You can see we get cyclic peptides, we have peptides with non-canonical amino acids. They're able to bind both to transferrin and to GLAST and therefore mediate more promiscuous binding if we want it. So we presented Peptune in the last few months. It's a very versatile framework. I'll show you why. So this works really well. By the way, it's the algorithm behind SOPIA. So I showed you how we were able to make fusion-specific binders. It's because we were able to incorporate specificity and affinity in the generation. And this, as you can see, is a very Monte Carlo tree-like algorithm. We've also used it for mRNA optimization. So in case that you have mRNA sequences, these are the four letters of of RNA, right? A, U, C, G. We now retrain the model on mRNA sequences, and we can optimize those mRNA sequences for properties. For example, half-life, ribosome load, translation rate, so that we can get multiple objectives while making this mRNA sequence, which again, people use for LNP delivery, for vaccines, and for other properties. These are the important properties you want. And we've shown that in experiments in our lab that when we use MR Nautilus, as we call it, we're able to get very optimized mRNA that express really well and are stable across long time scales. We've started a company called AtomBioworks on this so that now we're able to translate mRNAs into the clinic and to partner with companies that are optimizing mRNA for their vaccine or therapeutic applications. But the thing is, multi-objective generation, I've shown you all is based off this diffusion model architecture. It's not limited to diffusion, actually. We can apply it to another generative framework that we've been pioneering called discrete flow matching. And the idea of flow matching is that now you have this probability flow from a purely masked sequence. You see these M's here to the actual data distribution. These are the actual amino acids at those positions. And so we learn this velocity field that transports probability mass from a fully noised sequence to the correct sequence. And we can do this over all the data points that we have. This is really nice. It's just a different parameterization of the forward process. So we wanted to make multi-objective guided discrete flow matching. To do this, it's actually really a beautiful algorithm that Tong developed. We basically start with a random sequence that the model made. So the model has its own generative capacity. It makes a sequence. You can select a weight vector saying, oh, I think binding affinity is more important than solubility. And so you can specify the weights that you want for each objective. Then we transition each token at each time step. So for example, E at position 4, we can transition into different tokens, different amino acids, and we can score. Okay, we've transitioned it, new sequence. One, does it improve the objectives, right? We can score them on solubility, whatever. And does that new generated sequence align to the weight vector, right? I can improve on all the sequences, but am I offsetting the weight, the importances per property that I want? And so together, you add them together, and you get a transition score called delta S, which we then multiply by this beta term, and we actually edit the velocity field term to now be more cognizant of where it needs to go, where it needs to transport the sequences to get to these Pareto optimal fronts. As you can see, we can then perform Euler sampling to then say, okay, now we have a new velocity field, generate for me a new sequence that has a Pareto optimal set. Very quickly, you can see this works. Binding affinity, solubility, toxicity goes down, half-life goes up, non-fouling goes up. So we can optimize simultaneously all of these properties. And the beauty is we've actually tested this in the lab, and MOG-DFM, which we call it, produces therapeutically optimized blockers of Fc receptor. And this is done in collaboration with a company called Adaptive, and they tested 24 binders. You can see 1, 2, 3, 4, 5, 6 of them showed really good binding to FcRn, and you can see also that they have very good therapeutic properties. So we've been able to optimize across all the properties that we wanted. It's also the algorithm behind Moppet. So remember we had bind evaluator that I showed you? Well, Moppet is basically just bind evaluator plus an affinity predictor. We jointly optimize that and get these very motif-specific peptides that I showed to you. We're able to activate CARs, and we're able to bind to specific domains of proteins. But look, guidance loops, all this is done at inference time. I have a generative model, and I'm guiding it. But the thing is, this is really expensive. If you look at Peptune, its time complexity is dependent per diffusion step on the number of token substitutions per step, the number of branches that I'm expanding, and the number of rollouts, meaning the full trajectories that I'm computing. And so this gets really expensive. The question we've asked is, can we amortize this MCTS by fine-tuning the base generative model, not just using it and then helping it, but actually improving the base generative model with these Pareto optimal trajectories or these sequences. To do this, Sophia, this is her like third paper. She's an undergrad. The year she's made six already this year. Created this new model called TR2D2 in collaboration with Mole Tao's group at Georgia Tech. As you can see, the beauty of this model, you'll see it kind of roll out as it goes, is that we go and use the tree to generate sequences. And when we get to these endpoints, we score them with a reward function. We then hold the best trajectories, it'll come back, don't worry, in a replay buffer on the bottom right. And that replay buffer is what holds high reward sequences. So these high reward sequences are good ones. And then we use that to fine tune the base discrete diffusion model so that now the next time it goes, which it's going now, it will produce Pareto optimal sequences. So it's actually forcing the model to actually in the future iterations only produce good sequences. The model's distribution itself has been tilted. And as you can see, we do this by using this radon-Nikodym derivative. The idea is that if you have a reward, you can see the reward, it scores really well. You can actually update, you can apply the loss in fine-tuning with an upweighted weight such that if you have a good sequence, you want the model to really know it well. But if you have like a worse sequence, you have the model's weighting term for its loss being lower. So now we get better sequences because we are weighting the good ones in fine-tuning. By the way, TR2D2 is the state-of-the-art objective, multi-objective model. You can see compared to Peptune and unconditional models, we get much better binding affinity, solubility. Hemolysis is pretty good. It's still a little worse than Peptune, but that's fine. Non-fouling and permeability of these peptides are improved just by fine-tuning the base model. The ultimate goal, guys, though, is that instead of just doing targeted therapeutics where we have a target, we treat it, and we have a molecule bind to that target, could we instead isolate molecules that induce desired cell states? What do I mean by that? What if I could create a molecule that, if I put it into a stem cell, converts it into another cell type, like an oocyte? What if I could design a molecule that can put into a plant cell and converts it into, say, a nitrogen fixing plant cell? Or if I put these molecules into a cancer cell and I can convert them into a healthy cell, right? Could we shift the cell state? The problem, though, when I think about this problem, I think about it as in the form of a Schrodinger bridge. And the idea of a Schrodinger bridge, if you look on the right, top right, is it's converting an initial distribution, like cancer cells, to a terminal distribution, like healthy cells. It does this via learning a stochastic process that needs to conserve mass, right? You don't lose, the cell just doesn't die, hopefully, in the middle of this, and matches some reference process that governs the physics of this transition, or some reference process. But the problem is that for years people have done this, but they've not taken into account that the end states can be heterogeneous. Not all the cells are healthy. Some of the cells may be still cancerous or may have gone a different route. So to do this, we have trained this model called branch Schrodinger bridge matching. And the step one to doing this is to note that we have three separate or however many end distributions. This is the starting one. These are the three end ones. And so we want to learn the path between step branch one, branch two, branch three. To do this, we train separate bridges between the start and terminal distribution one. The start, terminal distribution two, start, and terminal distribution three. These are flow models that flow the probability mass from one to the other. And then in training, we jointly optimize all of these together to make sure that we minimize the total free energy of the system, we conserve mass so we don't just lose things randomly, and that the terminal distributions are matched, right? Because that's what the data says. So if you do that all together, we get this model called branch Schrodinger bridge matching. And as you can see, branch SPM allows us to enable trajectory mapping to heterogeneous terminal states. So that means if you have different terminal states, like this is LiDAR data, we just show that there's like a nonlinear path to these terminal states. We have to go around a mountain. And so you can see that we were able to learn those dynamics. But more importantly, in cell perturbation experiments, you can see very clearly, hopefully, that we're able to model branching from the terminal starting distribution to this terminal distribution, this terminal distribution, and this terminal distribution. So now we're able to model that in one network, can do that very accurately. But the thing is, in biological processes, transition states also matter, right? It's not just about hitting the endpoint. You also want to get the intermediate dynamics. And that's important in, say, cell differentiation or in protein folding, where intermediate points, right? Intermediate cell types, intermediate protein folding states really do matter. And so to fix this, can we use a Schrodinger bridge matching strategy to learn these intermediate distributions? And the answer is yes. To do this, we've created, Sophia again, created another model called entangled Schrodinger bridge matching, where we're learning a bias force that allows us to capture the dynamics interacting with the different particles, meaning I'm not just moving in a straight line from A star to A star, from A to A or B to B. I'm actually, these molecules interact with each other. And so we learn two networks, this R and V, that allows us to understand those interactions because these models are transformers. So this allows us to get intermediate dynamics from state A to state A to state B to state B. All I'll tell you very quickly is that this allows us to see and generalize to unseen perturbed cell populations, these intermediate distributions from start to end. And this diverges from the training distribution, which is good. We know it generalizes. And the beauty is this. Look at these proteins, alanine dipeptide, shignolin, BBA. These proteins also fold from start to end. And you want to model their intermediate dynamics. And as you can see, like you can see this very well with trip cage. You start here, you end here, and you learn the path. You're not going and randomly drifting off into these other areas. You learn the path accurately. So that's really cool that now we have this unified model for protein dynamics and cell trajectories, which is the future, I think, of AI for biology. So that's all. I think I left a little bit of time left. Thank you so much for listening. I apologize for going fast at the beginning. Hopefully, it was still understandable. We have a great team. The people I love to, I think all the first is I think I have the best students in the world. I'm so happy that they chose to work with us on these, with me on these problems. And these both teams work together so well. I'm really grateful for the funders that really give us the opportunity to try these. As you can see, a lot of our funders are for specific diseases, and that help because they trust that we can make a good therapeutic. So that's our job to do so. We also have great collaborators, experimentalists, as well as theorists who are better in theory than we are, and experimentalists who are better experimentalists than we are, working with us because we have this joint capability to solve these problems. We're really grateful to them. And to you guys, thank you for listening to our stories. I know there was a lot of them, but hopefully they were fun to listen to. I'm sorry I couldn't make it. The plane was delayed by a day, so we're really excited I could still give it via Zoom. And by the last thing I'll say, if you want to follow our work, we're in the middle of our fourth year as a lab. We just moved actually from Duke. We were there for 3 years to Penn. So if you want to continue following our work, our website is chatterjeelab.com. And try out our models. If any of these models are interesting, they could be used for biomedical data informatics as well. Try them out. They're on Hugging Face, so they're super easy to download and use. So yeah, cool. With that, I'm happy to take any questions with the remaining time I have.
Feel free to unmute and then.
Hello. Hi, I'm Patrick, a master student at DBMI. I'm really interested in your paperclip and PepMLM, so thanks so much for the great talk. My question is, with tools like this now able to hit proteins that used to be undruggable,
Yes.
what disease or clinical situation do you think that will be the first to show real patient impact from
Yeah.
programmable peptide therapeutics and why?
Patrick, it's a really excellent question. I think hopefully you saw that we really do care about that translation and that we believe that PepMLM and Paperclip while we were able to create great peptides to them, They didn't incorporate these other clinical features like half-life and others. That's why we developed Peptune and these other models that are now not only binding, right, which is what Peptune and Paperclip did, but also incorporating therapeutic properties like half-life, toxicity, and others. So I actually personally think models like Peptune, like MOG-DFM, and like TR2D2 will actually be the ones that create the molecules that enter the clinic. But maybe PepMLM and Paperclip will do too, but they're only incorporating binding. And that's only one part of the story. That's why, Patrick, we're doing this multi-objective optimization so that we can get to other therapeutic properties as well.
Thank you.
It looks like there's a question in the chat.
Yes, and I know Karash also has one here, but I can answer the chat one. I was Utah asked if you observed any practical issues of multi-objective, yeah, where the base sampling model limits it. Exactly. So Utah, you actually answered the question for me. The base MCTS, right? It's a generative model still, but the base sampling model may limit the exploration. So there's two things we did. The MCTS, we also added gumbel noise at the intermediate time steps so that we didn't just go down one path. We actually, if we got to like the fourth node, let's say the fourth time step, we actually added gumbel noise so that we then explored at the base. So we got some exploration. TR2D2 actually is very nice because it's now allowing us to adjust the prior distribution from the MDLM. And so now we're able to get multiple objectives to be optimized. And so and to be able to have a distribution that is much more exploitative, but also has previously been explored. So it's a really great idea. So thanks for that question. It's an excellent one. Maybe Karash, we could go to you next so that I can go to the rest one later.
Awesome. I appreciate it. Thanks so much for your talk, Dr. Chatterjee. Honestly, I was looking through the chat and my question is pretty similar to Maxie's. I was
Okay.
super interested in how your paperclip model was able to kind of rank and score these peptides. I
Yeah.
was just wondering, is there kind of what was the methodology between this ranking? Are you doing some sort of like similarity measure, or is
Yeah.
there what kind of is there any extra steps that kind of weight certain peptides more than others, or
Yeah, it is a similarity measure. The idea of CLIP, right, as you probably know, is it basically scores how similar the embedding of your peptide is to of the protein, right? Because we're basically training the model to learn those relationships. So we get a score out of it, actually. So because it's a cosine similarity, you actually get a score between -1 and 1, right? Because it's a cosine similarity between two vectors. And so if you're closer to 1, that means you're very similar. That means theoretically you should bind. But here's the kicker. That score is, remember how you train this model. While you train, per batch, you're looking at a subset of sequences. So it's not like you're looking at everything. So the score, the similarity score, is good, high, if you have closeness to your target and you have farness to the off targets. So in reality, you're both learning interaction and you're learning specificity. And together, that scalar score, that clip score, that contrastive score is a ranking of that. So if you have a score of like 1, then you're like, I'm definitely testing this. And we can literally just design peptides, we can throw them through, and we can literally get a ranking based on that. And it's really nice because that helps us to easily prioritize which ones we test in the lab.
yeah, that's super cool. Thanks so much. Appreciate it.
Thanks. Yeah, and Maxi, I think that hopefully answers it. It predicts binding and specificity, not stability, but if we want stability, then go to Peptune and these other models which incorporate other objectives. Great. Is there any? I can quickly answer Chuni's talk. Um, Chuni's wondering if for certain diseases would you need tissue or cell type specific delivery? Yes, the answer is so true. This is a huge thing that I say. We've used, as you saw, we use lipid nanoparticles to deliver them. That's fine, but lipid nanoparticles go to the liver 90% of the time. So that we would definitely need cell type tissue specificity because we need these peptides to work in specific cells. For example, the fusion oncoproteins. We want to work in the cancer cells. The problem is that if we utilize lipid nanoparticles, if we utilize AAV or these other tools, they naturally go somewhere. So my answer, and this is what I tell my students, I would rather have a simple molecule, like a little peptide or a drug that I dose and it goes everywhere, right? But the specificity should be at the cell level, meaning I go everywhere, but in the cell that I want to go to, that's where the peptide actually has its functional effect. And to do that, we have to be very specific in how we design them, and they should interact with its environment in the cell to be potent. That's, I think, the best. That's my opinion, but I think that's the goal that we should be striving towards.